everybody. Good afternoon, and thank you all for coming to the Interdisciplinary Humanities Center for today's event, which is part of Humanities Decanted. Humanities Decanted is an informal series in which faculty engage in conversation about their newest research and creative work. My name is Susan Derwin. I'm the IHC director. And before we begin, I would like to acknowledge the Chumash people, who are the traditional custodians of the land upon which the IHC is located. And I would like to pay respects to elders, both past and present. So today we will be listening to our colleagues, and then we'll have time for audience questions and discussion. Along the way, I should say, don't be shy about enjoying the snacks and the drinks. This is when we kind of graze and listen and enjoy in a very unstructured way. So if you get a little groggy, your neighbor will, just from the food and drink, it's going to be scintillating, though. I know you won't get groggy. So I must say it is my great pleasure to welcome our speakers, Bishnupriya Ghosh and Elena Aronova. Bishnupriya Ghosh is professor of English who teaches and researches on environmental media and global post-colonial studies. She has published widely on literary, cinematic, and visual culture, including her first two books, When Born Across, Literary Cosmopolitics in the Contemporary Indian Novel, and Global Icons, Apertures to the Popular. She also co-edited the Routledge Companion to Media and Risk, and today she will introduce her newest book, The Virus Touch, Theorizing Epidemic Media. And Bishnu will be speaking with history professor Elena Aronova, whose most recent book is Scientific History, Experiments in History and Politics from the Bolshevik Revolution to the End of the Cold War. She has also co-edited two collections, Data Histories and Science Studies During the Cold War and Beyond, Paradigms Defected. So we are going to start now with a short presentation by Bishnu. Enjoy, I know you will. Thank you.
Thank you, Susan, for the invitation, Erin for setting this up, and Elena for agreeing to have a conversation with me. We newly discovered each other, so I'm particularly grateful for you reading the book and engaging in this with me. So I was asked to do a very short overview, and so academics are not known to be short, but I will try to be. And you can ask particular questions when we get to Q&A. And what I wanted to do right at the beginning is to talk a little bit about the stakes of the book. Why write this book? I was writing this book for the past, 12 years when this new pandemic came along. So it was really about HIV/AIDS. And one of the things that has came out is the question of life, which is as a particular configuration of matter that becomes really valuable during epidemics. So there's a lot of debate in the environmental humanities about instituting that difference between the living and the non-living, and so on and so forth. Whatever the case, it is when life is in your shortness of your breath or the lifespan that you might leave behind, or really a sick animal in a wet market, whatever form of life you're thinking about, that configuration of matter is under threat, particularly because there's a new relation, right, that it has to engage with. And so the moment of the pandemic is at a moment in which we think about life as a distributed good, that you have the waning of the plant or animal host and the waxing or the flourishing of other kinds of life. And you have the species difference being instituted at that moment. And my starting point was, there are many stories about why I got to this very weird project from being a post-colonial scholar, but one of them is I began to wonder, well, how do we perceive epidemics, right? What is, and that's how I sort of stumbled on epidemic media as media technologies and forms that make infection legible. So one way of thinking about infection is fluctuating multi-species relations. You have more virus in your blood, less virus in your blood, so on and so forth. But the way we think about infection in terms of calculable distributions, you think of your PCR test, right? Tells you X number of viral particles in Y ml of blood or saliva. So epidemic media, and this one I all of us were watching for a while or during the last pandemic, it really makes this distribution of life calculable. It could be at clinical scale, your PCR test, but it could be a big epidemiological model that produces epidemic media like this, right? The reproductive rate of the virus and what that means for the spread of infection. So the epidemic always emerges. And I stayed with that idea of emergence for a while because emergence goes back etymologically to something new, but something that appears. And that is always mediatic. So one of the things that I was interested in is how epidemic media make infection legible, because this legibility has everything to do with whose life matters and how we value our lives in our communities and beyond. And so we often turn to the sciences, so many of us were following the research, right, to make things legible, to identify the problem. So I began to look at how scientific performances necessarily constitute biological agents as isolable objects, stabilized as such for study and situated within a well-demarcated surrounds that we would call an environment. And this is a sort of a classic thing. Some of you have seen this piece before. This is one of the animators I worked with, if I can get this to work, called Janet Iwasa, who did this animation, which is now called the asteroid aesthetic, right? So you have the HIV familiar virus coming into the cell, and it's kind of an existential struggle, right? It's a very classic edutainment video. So I'll just play it without
Step
sound.
one is
In the many histories of the pandemic that you see here, and let me go to this image, which is the image for the cover of the book. Right? And this is Luke Jerram, who noticed he made it in 2014 and 15. He makes these huge glass virus sculptures, they're kind of beautiful. And he had made variola, which is one of the biggest global viruses, before HIV. And then he modeled a future virus, which begins to look like SARS-CoV-2. Right? So it's interesting that he called it past, present, and future. And we have these sort of hoary protagonists like variola or HIV, SARS-CoV-2, when they come up, we sort of revert to a fear and loathing of germs. Even though we know by now, by the Human Microbiome Project, we all know some bacteria we depend on. There are some viruses, like in the placenta, has a viral gene. We wouldn't even have human reproduction without viruses, right? Still, the virus often appears to us as an eternal enemy, right? Against which we take a rather warlike stance. And this is a public health message, and you'll see what I don't even have to explain it. But basically, viral entry is always seen as a territorial infraction. We're always in a state of war with the virus. Okay, and this map can be, substituted by Uganda or any other thing. So, okay. So these are the kinds of epidemic media I started with, but that's not where I ended up. These are media forms, whether they were made for other scientists or public edutainment, are necessary for controlling infection when epidemics erupt as emergencies. The biological sciences are at the forefront in the making of epistemic objects. So many of us have followed the research in the hope of antibodies in trying to pin down the source of our infection or trying to figure out the origins of COVID. So what I did was I went to different laboratories where people were making epidemic media. And the scientists there were actually remarkably generous, explaining their method and the limits of their practice in the making of objects. So if you look at the second part, the second section, this clinical retrovirus lab, which has been actually processing HIV-positive blood since the 1985, so before the antiretrovirals, there you usually, the life form in question is the viral RNA or DNA. Right? And you kind of have PCR machines that read it, it isolates it, and then it counts it. So you render it as a media form. And so you have an object that is the viral particle within an environment, which is the blood as sample. And so I went to the lab looking for how then blood was processed, because my intuition was how epidemic media are made gives us another kind of story. So let me elaborate with the blood test. I'll show you a bad picture that didn't make it into the book because it was an awful picture. Anyway, but I was very interested in this because foundational to safety protocols was an implicit recognition of blood's unassailable transitivity. That is the capacity of media, now we're conscious of the air breath between us, blood or semen, to transgress the epidermal limits of the individual body. The clinical medicine lab, of course, is necessarily concerned only with individuation, the interior milieu of a patient's blood samples, and it stops at your epidermal limit, and it designates your body as the environment. But in fact, the safety protocols were an inadvertent acknowledgement, the gloves, the wipes, the hazmat suit, that there was an interior milieu's relation to the social and ecological milieux beyond the patient's body. So I came to understand these as nested environments, and they returned in the medical file. So it's demographic data on a group or a neighborhood, or as epidemiological data, co-infecting agents and mutations. And this was one of my research sites, were these smaller clinics that had been managing HIV all over the world. This is in Khayelitsha outside of Cape Town. And what was interesting here was you had the co-infecting tuberculosis bacterium as degrading or contaminating the pursuit for the HIV until they realized they needed to test for both. And if you see on the patient card there, the B is bloods and S is sputum. So now they test for both things, right? But what this told me, so these were the stories people were telling me. What this told me is there's this entanglement between something like a PCR, a machine, and these riotous biological agents when we search for the cleanest and most valuable signal. I only want data on HIV, but I'm getting data on many other things in that medium. The noise, however, that interrupts the signal illuminates the conditions of existence for the medium. This is Brian Larkin's point. That is from the greater milieu from which the sample is plucked. When we tune in to noise, error, and redundancy, as I do in the book, we apprehend something else, that other time and place of infection. So why at all do this? The apprehension, as I call it, unfolds very differently in each chapter. My point here is while epidemic media objectify to make a particular multi-species relation comprehensible. You only want to know about human and SARS-CoV. The very act of performing the processes of mediation that I watched involves sensing something else that is not amenable to capture, a relational sense of life, uncontainable to any one environment that we want to regulate. The degree to which that sense is palpable differs very widely during, in general, but during epidemics, one can't escape it. And this is where the humanities came back for me with the experiential sense of the epidemic. This apprehension, you've all felt it, is scarily diffuse sense of infection. It's everywhere and all the time, right? But it can also impel a relational understanding of life which underlies the environmental imagination. Imparting legibility but also a sensuous relation to the more than human world, epidemic media then are always environmental media. One of the main, you might ask, so what? Where does this take us? What about this experiential sense, this virus touch, as I call it? Why is it important to feel beyond the scientific objects? So I'll leave you with this. If you look at the structure of the book, chapter 1 is the big sort of historical chapter, which makes a historical claim about this moment that we're in. But chapters 2, 3, and 4 are all about scale. Right? One is about molecular, one is a clinical scale, and the last is about a planetary scale analysis. So implicit in the argument, in the structure of the book, is a reading of epidemics as biological and ecological, social and political, non-linear and multi-leveled. Acute infectious disease emergencies are not just medical emergencies to be fought with medical solutions, vaccines and therapies. I'm not an anti-vaxxer, I know how life-saving those cocktails have been for my generation of HIV/AIDS affected folks. I know how life-saving the vaccines were. But without devaluing medical solutions, what I'm after is this war on germs, right? The point is not to let the short-term, expensive, and habitually inequitable solutions, and we know this from vaccine capitalism, obscure the longer view. The pandemics will keep coming, history returning as farce, until we articulate human health, animal health, and biodiversity together. So, of course, the statistics have been out there since the early 21st century. 60% of viral emergences are cross-species spillover, zoonotic spillovers. 71% are human and wildlife contact, right? So this is the large picture. So the very constitution of one form of life, this object, this isolable virus, cuts it off from its relational distributions, and this problem solving that creates a target that you can apply a medicine to kill it might generate unintended consequences. And some of the unintended consequences for me was obscuring the longer fight. But it is utterly difficult to keep this multi-scale, nonlinear perspective at the forefront during the kind of emergency we've been through. Just think that how we'd rather believe the lab leak theory, because it places the blame safely in human hands. Much harder to think of large-scale structural changes, right, that has to be done, that have to be undergone in order to change human-animal contact zones. So I'll close here. Epidemic media, for me, is the key to this reorientation if we are to have livable pandemic futures. Media, as substrate, as apparatus, as infrastructure, can generally foster productive apprehension. But epidemic media generate an open-ended provisional form of knowing multi-species relations when they feel most difficult. That is, we really don't want to have a relationship with SARS-CoV-2. This is what Isabel Stengers actually names the new memory for unfolding catastrophes. She's talking about fires. She says we've had this smoke in our nostrils for a long time. And now's the time for a new knowledge paradigm, right, for the future to combat this. My gamble in the virus touch is to refract our analytic gaze from self-evident media forms, such as the viral image, and to consider what the processes of mediation tell us. This making, doing, acting that discloses what epistemic objects keep at bay, this making, doing, acting that places us always in the more than human world. And I've only touched on one chapter, of course, but I'm happy to talk about the others because they do different things with this constitution of objects. I'll just stop there. Thanks.
Thank you so much for this wonderful presentation, for writing this book. And the first thing I really want to say is to congratulate you on the book.
Thank you.
I really enjoyed reading it, and I appreciated the opportunity to engage with it. So thank you for inviting me as your interlocutor here. So I have a number of questions, and I don't think that I will get to all of my questions. So I really look forward to continuing conversations afterwards. So my questions are coming from my perspective as a historian of science, but I want to start with a question/comment that comes not from my kind of experience and my perspective as a humanities scholar, but rather from my early experience as a scientist. And yeah, I know you know the story, but let me repeat it again. And I will explain why I invoke it now. So I received a degree in molecular biology and biochemistry in Russia in the 1990s, but was a very interesting and difficult time in this part of the world with economy collapsing after the collapse of the Soviet Union. And I have taken a job in a clinical laboratory that was just starting to use the then relatively new PCR technique that we are all familiar today to diagnose a host of different viral diseases, including pretty serious ones such as hepatitis. And the HIV/AIDS was on the horizon when I started working there. And so working there amidst this the collapse of economy and the collapse of the entire supply infrastructure for scientific research, we lacked very often some basic lab supplies, such as microtubes for sampling. And as a result, we almost never, for a short time when I worked there, we almost never had a clean negative control. And that means, you all know that PCR is a very sensitive technology that indicated that we had a contamination, and this contamination basically should have invalidated the results. So the reason I'm telling this story is that for me, and especially after reading your book and kind of returning back in my memory, so for me, back then, seeing those unwanted traces on the electrophoresis gel, where there was supposed to be none, was my experience of the virus touch. The virus touch in the sense that you use it in the book and kind of in the title. So I was trained, so let me unpack that kind of from my experience from this story. I was trained to work with viruses as standard model organisms. Okay. So I was trained to work with viruses as model organisms, that is to say, standardized and controlled, designed, made to produce reliable and replicable experimental knowledge. So seeing those unwanted traces was experiential experience. It was this kind of another kind of knowledge, as you call it in the book, referring to the scientists that you worked with, interviewed, and participated, engaged in participant observation in the laboratories. So what I getting at is that, for me at least, from my kind of perspective, there was this sort of uncanny parallel between my experience of the virus touch and the experiences of the scientists that you followed, the scientists who worked in this urgency of another kind of crisis that is and was epidemics. So it seems to me, and that's what I was getting from the book, is that those scientists who worked in those emergencies of health crises are more attuned, perhaps, than others to the limitations of scientific technologies, of different technologies, experimental technologies and experimental knowledges of the virus. They are perhaps more attuned to the uncertainties of the knowledge of the virus. I'm losing my train of thought, so let me just look at my notes. You call, so those scientists were, in other words, attuned to this another kind of knowledge. right, experiential, kind of central, right, sort of, I would say, local as well, a kind of flip side of experimental knowledge that is generalizable and abstract, and I would add global, knowledge that can travel, right? So this interplay between experimental and experiential knowledge that I found so fascinating reading your book, or as you call it, the interplay between thinking and feeling, as it's being played out in and through media. So this is one of the themes running through the book and captured in the title that I found extremely compelling and extremely fascinating. So the question that I want to ask you is actually about the readership for the book. I realize that your primarily kind of primary intended audience are the humanities scholars. But at the same time, since you followed many scientists and since you worked with them and kind of engaged with their work, so I was wondering, do you see a possibility for a sort of feedback loop here? That is to say, do you see scientists or perhaps biology students among the readers of the book? And if so, what key lessons your book may offer them?
That's a hard question. Yes, you're right. When I started to work on this, it was a humanities project to begin with. I was looking at cultural texts, huge archive of HIV/AIDS media, and I sort of began to look at how this constitution of objects, and I kind of fell into science, and I was there for 10 years. I looked up, and it was 10 years later. And then this other kind of knowledge, I came around back to thinking that this experience that scientists have, and I think I want to stress that they always have it, and particularly in the urgency of the epidemic. Yeah, this experiential sense that scientists have, the humanities have a very long tradition of being able to deal with that. The experiential. We have methods. We have ways of characterizing and documenting it. And so, for instance, the many stories that people told. So a lot of the stories in the book, in the labs, are really about, one story in the blood part, right? They were so kind. One of the clinicians had taken her own blood the day I landed up to watch the PCR cycles. And they were like, we took we tried with a sample of blood, but there was so much virus in that blood, it could have walked on its own. And it was like a, whatever comment. And I was like, do you mean it was multi-species? She's like, no, we don't think at that level in the lab. And I'm like, why don't you? So there were all these. And then I began to think that comment, the fact they were thinking of species and specimens, the blood specimen, was actually not important in this lab, which had to, in fact, deal with the clinical scale. So one of the methods was actually thinking about what people was telling me, the stories of how they came to make blood, right? That for scientists I found were recessed, but that I had a different perspective on those stories. So that's for the humanists, in terms of methods. The question you're asking is a really interesting one, because I found some scientists actually are theorists of this other kind of knowledge already. So Anne Lodiso, one of the people who I track in Uganda and DRC, she goes looking for animal hosts, basically primates. She was very, very already tuned into this, right? So she would take fecal matter and she would look for viral strains, but she would also look for seeds and insects in the fecal matter, because that tells you about the habitat, right? The virus is never just the DNA or RNA. It's this entire world that it carries with it. So she didn't think of herself as theorist, right? When she said, so I was like, yeah, but you're attuning me to an environmental imagination. So I think your question makes me think perhaps scientists who are often have to be careful about the limits of their practice, as Isabel Stengers has said, and do not make partial connections because, that is against expertise, it's not deep enough, it's, exactly this book, right? It's not anywhere, right? To make those partial connections, to me, is the environmental move. And maybe, systems biology that looks at complex living systems may be close to it. I don't know how much of, so I see even people who are processing blood in a clinical retrovirus lab devoted, the most anthropocentric of places, are in some ways intuitively environmental. So I think that is one of the strong arguments that I make in the book, that we're always actually thinking across practices and disciplines, and that is very much a part of the environmental imagination. You can't do it within one discipline alone. Yeah.
Precisely, and maybe not only environmentalists, but in disguise humanists as well. So just sort of to kind of return back to my experience, for me this virus touch experiential side of knowledge, it raised questions that I have never thought really much before. Well, most of all ethical questions, given the stakes of the diagnosis we were given to people. But I basically left the lab and the molecular biology altogether and went on to pursue a PhD in history of science, starting to ask different kind of questions about why and how do we trust data and why do we trust those results, kind of meta kind of questions. So it just struck me that in those crisis spaces that you're looking at and practitioners working in those spaces, they are much kind of, as you say, intuitively environmentalist and kind of humanists. So we are in the interdisciplinary centers.
May I add something else?
Yeah, sure.
Yeah, I think that, the other part of the book that you just saw a brief glimpse of is Kailich, the small clinic in Cape Town that I showed. I was interested in talking to you precisely because you bridged the two. But the book is very much devoted to these small points of care all over the world, particularly in the global south, where people are actually theorists of the virus touch. They work every day with people who's, they can't come to get their meds. It's been too much rain. There's water logging. There's no electricity. And this is all in the medical file, right? The entire habitat. And they are the ones who have delivered, if HIV/AIDS control has been a success, 67% of the HIV-affected people are on the meds, it's because of them. So the value chain always comes back to the lab. Right? The story of AIDS is 1995, big scientific discovery, thank God for my generation, not undercutting that, but that is the triumphant story. And so part of it was also, speaking of people who live in the urgency of the epidemic in the global south, things are still at high crisis. So those people, I wanted to actually tell their stories alongside the labs on an equal playing field. So part of it was that. So, yeah.
Thank you. Well, just going off from there, so much of your book is about collaborations, about collaborative ventures between, I especially kind of found interesting the prominence of collaborations between scientists and artists in creating those visualizations of the virus. So it's really collaborative art-science enterprise throughout, right? And it makes sense, speaking of the media, epidemic media. So you call this transdisciplinary travelers the modern practitioners. So you explain that as modern practitioners, the scientists featured in this book collaborate with artists, software engineers, writers, healthcare workers, and local animal experts, and so on and so forth. Reading your book, and maybe I'm mistaken, but I could not help but noticing that many of those modern practitioners, people working in the emerging fields, on the borders of established disciplines, many of them are women. And so I was just wondering, is there any gendered dimension to the story?
That's also an interesting question because it links, I think, my epistemology is to think of difference as constitutive, whether it is in the value chain, healthcare activists working on the fringes, and scientists having everything in their lab. My epistemologies have always been feminist, but I didn't start there. I was inspired by feminist queer work in the 1980s and 90s, which really talked about how to write, Paula Trickler's work, how do we write a theory in an epidemic, right? She was writing about HIV/AIDS. And so the book ends as a kind of an acknowledgement of that, saying how to have media theory in a pandemic, right? So there is a very direct reference in this urgency of the epidemic. How do you theorize? And a lot of it is, of course, I'm very interested in people who just take their drugs on time, or caregivers. It can be your lover or your friend or family or whoever, right? I'm interested in those kinds of circuits. And I don't want to gender them necessarily, because, of course, I've seen among ACT UP activists, how it works out. But some of it I fell into through actually research. For instance, the image chapter is about two women, Donna Cox and Ellen Sander, whose initial virtual environments that they created in images of the virus ended up being the model for the cave, which is the virtual environment, the cave virtual environment thing that is so big now, and everybody wants one in the lab, right? So there are unacknowledged legacies. So I would say my work is always feminist in terms of its epistemology and the things that I've learned how to approach things in thinking about difference and thinking from the other side, And the second feminist lesson about epistemology is that to work in the master's house, you need to know the master's tools, right? Both post-colonial and feminist studies have taught us that. So I learned the science. I took classes in biology, right? It's a key lesson.
And you made your own choices of whose voices you wanted to feature in the book. I suppose that's part of your feminist stance. So I was actually thinking about, and sort of just to give you an example from the history of science, an interesting parallel might be the early history of genetics, when it just emerged as a new science, new scientific specialty. And some historians of science noticed and made an argument that this new scientific specialty was an opportunity for many women at the time who were able to make successful careers in this new field rather than in such established fields as zoology and botany at the time, with higher barriers for women to pursue careers in those disciplines. So it's just a kind of another aspect to the possible gender side of the story.
Jennifer Doudna's work on the mRNA vaccine. We wouldn't have it. Yeah, she was the person who got us the mRNA vaccine. So yeah.
Now moving to the core argument of the book. or at least one of the central arguments the book is making. Your articulation of epidemic media as this kind of crucial site, for the better, for the lack of the better word, or space for recasting global health crisis as social, ecological crisis and catastrophes, and for directing us towards kind of new multi-species politics of health. And kind of teaching us also, I suppose, how to live in and live those multi-species entanglements. So my question is about what you mentioned in the presentation, and it's also a kind of a statement in the book that epidemic media is necessarily environmental media. And so I was thinking and wanted to ask you, what is then the place of epidemic media within this kind of larger space of environmental media that are probably playing the similar role? So I was thinking in particular about, what might be called radiological media, with Chernobyl as a kind of epitome of radiological disaster, by the way, happened around the same time as HIV/AIDS epidemics erupted. And now this kind of, we can see some parallel developments of within the media, Chernobyl art, playing a very important role at sort of articulating both, those kind of enacting those different spaces, like, for example, using the soil from the exclusion zone to grow plants in Britain, and then to expose the photosensitive paper with those plants, creating those radio kind of, not the photographs, but this absolutely fantastic kind of traces of radiation. And there is this interesting parallel and similarities between virus and radiation. Both are invisible, both are only legible through those different scientific technological media, right? And both invite us to think ecologically and multi-species, kind of invoking those multi-species politics and consciousness. Another example is climate change research, and the Scripps Institution of Oceanography, a neighboring institute of one of your sites. So it regularly invites and hosts artists to articulate, you cannot feel global environment, you cannot feel sort of global climate. We need mediators, and that's where artists come in. So all those different environmental media, so how do you see and how would you place epidemic media in this kind of broader spectrum of environmental media?
Yeah, I would answer that into, thank you for that example. I think it's a very good one to think at the molecular level of what's happening with radiation and what's happening with infection, right? There were two reasons why I make that claim. It's more or less throwing down the gauntlet of sorts. One of the reasons why I call epidemic media environmental media is because every time someone says biology, right, we end up saying, oh, it's naturalizing, and it's the one really anthropocentric science, only concerned with the living and only with life, and look at all the damage that is done. Somehow we get to National Socialism whenever we talk about biology. Well, I was trying to think with biologists about how biological sciences, and UCSB is a great example, they're huge, right? They're many things now. And they have always been aware of the fact that Stefan Heimdijk in MIT has this fantastic essay called The Limit Biologies. And it talks about, the ways in which there are biological organisms in space, or you have extremophiles, and the virus is always considered the fourth form of life. So chapter 1 has a huge history of the virus, in which it is the living and non-living, right? It's precisely the point to think about the biological and geological together. You cannot only think about it biologically, even though you do that pretty often. And so the virus is living because it wants to reproduce, right? And it is irritable. If it senses a host, the viruses talk to each other. It's called quorum sensing. And then they jump into the host. But it can't metabolize itself. It needs our resources, right? It's a parasite, right? So it doesn't have one feature of life as we know it, right? So it's a limit case. So if you really think from the virus, right, then you're forced to think about the biological and geological together, that we live in an age what Hannah Landecker has called anthropogenic biology. So much of industrialization has happened that whether it's our food or our waste, we are no longer, you can't separate. She calls it, we live in dysbiotic times. Our biology has changed forever, right? We cannot think without chemicals or industry. We cannot think outside the planet. We cannot think only about our bodies, right? So I was really going after the anthropocentric, spinning biology to anthropocentrism only. And that was sort of one of the main drives. And I think radiology is, again, there was a second point, which now I can't keep in my head, it's too late. But essentially it was really to force the equation and think about the biological sciences more broadly. Yeah.
Thank you. So my next question comes from my own interest in Cold War scientific spaces. So I have to ask this Cold War question. And of course, Cold War is not brought up in the book, but I think it's could be an important part of the story. So I want to give just one example, which is most tangibly related to the story of HIV/AIDS, which is at the center of the book. And this example is the history of Cold War era massive collection of blood samples from people in colonial and post-colonial spaces whose blood was drawn and then beginning in the 1950s and frozen using then relatively new technology of refrigeration and then stored in biobanks in the US mostly. So it was a time of proto-environmentalism with Rachel Carson's Silent Spring, and everybody was concerned about ionizing radiation and about pollution. So indigenous people, in particular, were excessively sampled and studied, not to help them, but to create and sort of save for future a sort of frozen kind of snapshot of what was considered less polluted bodily evidence in order to use that in future to manage future health risks of increasingly polluted atomic models. So, beginning in the mid-1980s, this blood collected from 1950s through 1970s and stored in the US was thought and assessed for the origin of emergent infectious diseases, including HIV/AIDS. And in one celebrated story, I'm not sure if you are familiar with this, but that was a kind of one of the triumphs of cryobiology,
one sample collected in 1951 in Belgian Congo tested positive for HIV, and that was the oldest trace of the disease ever documented, and this was revealed through this Cold War-driven accumulation of blood. So Joanna Reading, the historian of science, tells the story of Cold War origins and subsequent reuses of frozen blood in her book, Life on Ice. Going to my question, I actually don't want. I'm not going to ask you about the Cold War. I want to ask you about something else, because you are telling a different story, and of course, everyone when knows that at some point, writing the book, we need to make choices, what to include and what to leave out. So I wanted actually to ask you about the choices that you made, and in my case, at least, some of those choices and decisions. No, that doesn't belong to the book. I want to make the book manageable and kind of focus, so I have to leave it out. And some of the choices are really painful because I really kind of love that. So I was wondering if you have that kind of material that you collected and researched for the book that didn't make the cut.
Oh my God, that would be opening a whole closet of things. Yes, so there was a lot. One of the things that the Cold War did was there was so many fabulous stories of, like, it always became about biosecurity. The RLS event where, someone was weaponizing smallpox, and I just got sick of the war thing. So I was like, no, I'm going to look at this, our relationship with viruses somewhat differently. So, and so this blood sample thing is a wonderful story. I wish it would have made it into the book had I known it. But, so there is a genetic tracing of HIV back. It emerged in the Cameroon about 100 years ago, right? And some of it was endemic, some strains. It's only the HIV-M group that became the global pandemic, partly because it was near the Kinshasa highway, right? So it was global transport, etc. So there is this kind of viral genetic tracking that, and the 1951 sample would, I wish I'd known that. That would have made it into the 59. Oh, then we're on the same date. Yes, I have the 1959. But, and then, of course, 1980s is when you have the symptomatic recognition. Something's happening with these folks who are getting this syndrome of infections, right? Yeah. So that's what I'd say about the Cold War. The reason why I left that out is because there's a lot of fabulous and important work going on biosecurity, and I didn't want to be a part of that conversation because then you're critical of biosecurity and biopolitics very, very importantly. But what is to be done? And I kept remembering Lenin, if you will, on that question, and thinking what is to be done? How can the humanities, with its enormous archive on imagination, really think beyond this anthropocentric mode of war, right? And so the story I tell is really in terms of the US, right? And not only the US, but the WHO, the declaration of the war on germs and the mass manufacture of penicillin during World War II, which sort of ends with a triumphant note in 1978 when we're saying, oh, smallpox has been eradicated. And then in 1980, you have Hanta Marburg HIV burst on the scene. And there's a real sense of the loss of the war on germs. And Melinda Cooper, who writes the book Life as Surplus, has already done a fabulous book. She's already written it. So I was interested in how suddenly at the end of the 20th century, it's what I call the epidemic episteme, there's a real melancholia. We have lost this war. And if we have lost this war, if viruses will keep coming, how do we deal with it, right? So in other places, lots of things falling out of the book, like EcoHealth Alliance, these environmental folks that I end up with the end, They've been working on, say, simple solutions, right? We've destroyed the habitat of these monkeys. They're coming into Bangladesh, into the streets. Let's build a buffer zone with fruit trees that the monkeys can eat and humans can also use, fruit sellers can use. There's some very simple on-the-ground solutions about land use. Now, this is something that we can do if we understand viral emergences to be not about just DNA and RNA. So it was a little bit, I know it's a little soapboxy, but the choices that I made told a certain story which went against the war on germs from, the post-World War II dream that we're going to kill all the germs. And it's only yielded antibacterial resistance. It's only yielded pesticides and cancer, right? So we need to think beyond these industrial solutions. And that, I was pretty passionate about that. So I kind of left the war things a little sidelined.
That's a wonderful place to open up.
To be continued. Thank you, Elena. To be continued.
Raise your hand and the microphone will arrive.
It's really not a question, but more of a comment in line with the last question. I was just thinking of if the war on germs misses out on the irrevocable propinquity of things, and has this fantasy of kind of making a cut there. Similarly, the war on drugs, Nancy Reagan's famous war on drugs, forgets about pleasure. And that's the problem. It never becomes a part of the equation. The war on terror forgets about what causes the terrorism in the first place, and as if that doesn't matter. And the war on piracy now, which, well, I'll leave it at that. Media piracy, I mean.
Comment?
Oh, I'm against all wars. I think if anything we've learned from HIV/AIDS affected communities is that if you leave pleasure out of the equation, and if you don't recognize sort of a politics of affection rather than of antagonism, then the battle is already lost. So yeah.
Thank you. Thank you. This is absolutely fascinating. And I have a question, I think, a bit out of left field. Of course, I haven't read the book, but I was struck by the terminology of war and of a more, let's say, universal or cosmic appraisal of epidemics. And so my question would be, is there a connection or a continuity with an early, very early paradigm of epidemics, which is the story of the plague in Thucydides, which is for Europe the first story of epidemics. And thinking about it, it seems to me that, of course, you have the war, it's the Peloponnesian War, and it's also an image of the breakdown of society, the breaking down physically, biologically, but also politically. And the king, if you like, is about to die as well. And then you have, the presence of animals, zoonotic transmission, because refugees from the countryside are in the town bringing their pigs and their animals, and they're all cramped in there, and then the rats come, and and then it starts. But really, it's not a one-time thing. It's sort of a cosmic or cyclical understanding that war will always be with us, and it's just one of these things that recur, and like seasons or like regimes, and epidemics as well. So I'm wondering if that paradigm is still useful for your thinking, or if there's really a break, a paradigm shift, if it's no longer, or we're no longer living with it, and if so, what replaced it, or how did this break happen?
Thanks, Dominique. It's a complex question, and I'm seeing many chairs who made time in their day schedule to come. So thank you also for that. I think what you're describing as a classical wars are contact zones. So all kinds of things happen. Populations sometimes not in. If you look at 16th century Mexico and you look at the wars that went on there with the Spaniards, right? You have the flus, etc. We know, and smallpox, right? There are 3 outbreaks of smallpox, 16th century Mexico Valley. Anyway, so wars are contact zones in general. That stays always. But I think what I would say is we have actually returned to this more classical moment of thinking about cosmological is one, but also to think about this kind of multilinear way of, non-linear way of thinking about infection. I think the difference was germ theory in the mid-19th century, because germ theory basically said, here's a microbe and here's its medicine, right? And this is the causality. So it actually, this kind of singles out, that there is this antibody, this treatment. And then all through the 20th century, we've done this until we lost the war on germs, right? And then there is a sense, okay, we can have the medicines. They're going to be expensive. We're soon going to be paying for vaccines, right? And some parts of the world in India, people are paying for vaccines, right? So they're always going to be expensive. It's always going to hit the vulnerable communities the most, right? So instead of doing that, if we have a larger sense, that was actually there in the classical period. So I would think actually from what we call modernity, say the 1700s, 1800s, bang to Pasteur, right? That's the moment in which we have a shift in the causality. It's a linear causal approach to disease that I think has done us a great deal of harm. So I think the classical texts are really good to reread at this point.
Thank you, Vishnu. So I had a question regarding the idea of deep time. I'm asking because deep time because you're talking about scale and geological, and I could not help but think of this example that as the ice cap melts, there are dead bodies of animals containing viruses that we do not know that will unleash on our. So how does virus touch keep us thinking about the deep time in different ways in light of environmental media?
So another great question which links, I often use the term biogeological churn in the book, because it's these things are enmeshed in each other. And I would answer your question in 2 ways. There is the immediate urgent emergency that as the permafrost melts, there are more viruses going to come, right? So this is a climate problem, right? And there is somebody called Penelope Boston, for instance, who she's an astrobiologist, right? Who goes down into these caves and she looks at these big virus crystals. Like they're just like lying there, right? And then, of course, if they sense a host, who knows what might happen. But, so they're already in the Earth's matrices. They're already there, yeah? And the thing is, and this is why I think entanglement doesn't mean go hug an animal. I think there are literally, we have to think about the people writing about flourishing and awkward creatures and how you actually you want to disentangle from them, right? So I think that's why I call it a multi-species, not a relation, but a politics, right? That there is a political distance between viruses and us. So there is one trajectory which is about things lying under the earth that are coming to the fore. The other history, I think, is Lynn Margulis, right? Who is amazing in thinking about the virus as something with an evolutionary grit. It's an ancient piece of RNA, right? Which has been in a 4 billion year primordial soup, right? A pre-Luca cell, right? That's still, but it became a parasite at one point, right? So there's another deep scale history of viruses and where they came from, a geological history, right? That I don't have the competence to write, but I draw on her work and the imagination of Gaia as a big old microbiome, right? That we ignore at our cost. Yeah.
Yeah, this question just occurs to me now in this thread of conversation. But you talked about the biologic and geologic, but in a possible other term than the geologic is minerality or the mineral. And I wonder how this enters the picture, as you describe these crystals under the earth that contain virus particles. But then in the science side, I wonder also how the mineral comes in. So protein crystallography, but then also the way that blood carries all this iron, or how certain tests for blood also really hone in on certain mineral components as they're suspended in fluid. And so there's a kind of I'm sort of wondering like what might be at stake in theorizing the valences of the mineral compared with the geologic as different heuristics into these kind of proximities and thinking about stasis and movement and these kind of edge cases you describe, which for me also evokes other edge cases like corals that also have, of course, that mineral biological kind of interaction together. So I think the virus also kind of constellates with some of these other figures, but I'm just curious about your thoughts on the mineral.
Thanks, Melody. And also, I owe you thanks for my conclusion. There are so many people in this room who have helped with this book. I can't even name all of you, but you're certainly one who've been really inspiring in your work. I'd say two things. One that I don't know much about, and the other that I do. One of the things that is important for modeling viruses is this crystallography, right? That you actually, you take a crystal and you, through optical mechanisms, you actually look at the structure of the protein, right? And this is really, really important for viruses because, in terms of viral entry, because say your cell is made up of, this number of proteins. And as the proteins morph and shift, there are different grooves on your cell. This is a very banal way of putting it, but anyway. And viruses, like HIV, for instance, they often talk about they go in for the T cell because they can fit into that groove, right? And part of the blockers, the medicines, try to block the groove. These are the protease inhibitors, right? They inhibit entry. And the virus, of course, mutates, and it tries. So seeing the protein, its structure, how it makes the groove, is very important. So the history of minerals is actually central to understanding the virus as a kind of a mineral form in that way. So that's one. And Natasha Myers, of course, has that wonderful book called Rendering the Molecular that takes a deep dive into this process. The other thing that I don't know very much about, but a friend told me when I was working on this part of it, saying, well, what's happening if you look at people's urine samples for virus, you'll also have mineral deposits. And so I'm like, and then what about them? They said, well, there's a relationship, and then it got so complicated, right, that I couldn't follow it. In fact, the first structure of the book were the three sciences of the virus: virology, immunology, and epidemiology. The immunology was so complicated that I actually had to drop it. I'm like, I just can't get my head around this. So then the mineralogy part of it, which is also very important to the virus as a partly inorganic thing, was something I couldn't understand. So it's not in the book, but it's an excellent question. I think there's a relationship there to be tracked. Yeah.
Thank you, Vishnu, for that. I can't imagine how daunting the task must have been to break the barriers coming from humanities. I mean, I can't wrap my head around it, so this is amazing, and I congratulate you for this wonderful work. But I want to take you back to your discipline of literature, in a sense, and ask you this question that, multi-species relationship that you kind of conclude with was something that was done through mythology earlier. And I'm thinking about Amitav Ghosh and, talking about Sundarbans and Bon Bibi and so on and so forth. he also, in The Great Derangement, talks about the failure of literature, in a sense, to take on board a different way of thinking and writing, right? Different kind of literature that can be written that takes into account our maybe climatic crisis or ecological crisis and things like that. Now you have actually, broken a barrier coming from humanities and, the social sciences and thinking about what we very often leave to the scientists, in a sense, right? So in terms of storytelling about this crisis that faces us, have you thought about how we can tell these stories?
I know you're the expert on storytelling, so anything I say will be banal here. But I think about Ghosh, of course, has talked about literature as a set of stories and practices that we value as literature. Yeah, and in that way, I'm very literary. I'm very suspicious of any value system, right, of what we ascribe to what we think to be literature, because literature is, a very wide bench for me. It's an imagination. It's also an epistemology. My close reading comes from training in literature, right? So, but to your question then, one of the things that I've been very interested in, goes back perhaps to Dominique's point, is that, for instance, in these virus stories, like there was, in smallpox, in India, for instance, you worshipped the goddess Sitala, right, who was known as the Cold One. And she was the goddess of smallpox. She was readopted as the goddess for HIV in some parts, right? So she's the virus goddess, right? And when you worship Sitala, you had cold curds, you had neem on the sores, right? So now if you put some cold things on the sores, those flakes are not going to fly about and infect someone else, right? So it was a very holistic system of health, actually, that if you read about Sitala worship and the stories people tell about worshiping this goddess, which to me is literature, right? You actually get a kind of a health diagnosis and health management, right? So I think those stories, and then, smallpox varialization, this history, was very widely practiced in China, India, as well as Turkey. But it was only when Edward Jenner in the 1700s does the vaccination, because he notices that the milkmaids who attended cows and were in contact with cowpox didn't get so sick from smallpox. That's why it's called a vaccine, right? Vacca, from the cow. Then he invents the vaccine, right? And then, of course, everybody wants a vaccine, and it's no longer varialization, right? So there are stories and stories and stories. But your question makes me think, goes back to Elena's question, which are the ones we want to tell? So now I think there's a collection called Pandemic Histories, the journal History of the Present has brought out a volume. And there I tell these stories, variola stories, and I choose 3, all of which have to do with children who were actually absolutely key to vaccination. James Pipp was a servant boy who was experimented on, right, by Jenner. There were 3 children who carried the vaccine all the way from England through Baghdad, through Turkey into India. And they were all mixed race children, right? So there's a history of children to be told. So I've told those stories of people who actually did the hard work, but they kind of fall by the way, historical. And you know this very well from your minor histories.
Hi, Vishnu.
All right. Well, okay. Last question.
Okay. Can I ask a two-part question?
Yes.
Sorry, I'll be quick. Well, it's not actually 2 parts.
course,
It's 2
sure.
different questions, Vishnu. Sorry. Congratulations. My first question is just a curiosity. This book started out as a HIV/AIDS book, and then the pandemic happened. So I was wondering if you could just say a little bit about how the book shifted in its conception once the pandemic hit. My second question is as you were sort of describing the body as a multi-species biome, you had to do this kind of apology and hedge where you said, now I'm not against vaccinations. And I'm curious about that moment and what it says, because in part, sometimes the language of the body as a microbiome has been weaponized by the right and by anti-vaxxers to say, right, what we need is a herd mentality rather than a vaccine. What we need is not to shut down the economy, but to allow circuits of capital to continue going because we just have to adapt. And then all over Santa Barbara, people are proudly sort of proclaiming that they refuse vaccination. So I'm just wondering if you could say a little bit about, this, the political outcome of the kind of narrative you're helping us think about around kind of multi-species being, and how we might think about the ways in which it then kind of gets weaponized by the right.
It's a great question. I'm glad you asked it, because one of the difficulties was I was working on HIV/AIDS and global distribution of risk, and the whole thing about the pharma, etc., and very firmly in critical global health studies, right? And one of the difficulties I had in the book is to marry critical global health studies, Paul Farmer and other people who were talking about inequalities in thinking about infection, who dies, at higher rates. That was always, I've written a series of things about it. To have that alongside this planetary health thing, right? Which seems, where the human seems to be one abstract thing, right? Rather than differences within human communities. So that's why the book actually says that you can't think of this as biological and ecological without seeing the social and the political, right? Alongside. It is very hard to, keep together. The way in which I deal with it, in the question you're asking about how the right wing has mobilized this, I think of the microbiome as basically I'm interested in a multi-species politics. Yeah, so that multi-species politics in that chapter 1 talks about the huge activist mobilization around HIV/AIDS, right? Which said we actually need to talk about the most vulnerable communities in an epidemic, right? And it's not a competition, between animals and humans, this is an uneven terrain, and we have to think wherever there is difference instituted, we need to look at it. So that's why in that chapter, it's an uneven chapter which talks about the history of planetary health and the history of critical global health, yeah? And the unequal relations between us and other species, unequal relations within our communities. And both are actually the motors of the book. So I don't think reading this book you could come out as not an anti-vaxxer, of course, that's not, although I have my own problems with medicines, but not an anti-vaxxer, but I don't think you could come out without thinking that willed biopolitical neglect you look at what happened in India, right? 1 million migrants didn't have a place to go, and something I've been writing about in another set of things. But people just died because, they didn't have a home or a place to go back to. So that willed biopolitical neglect is also part for me of multi-species politics. Some vulnerable communities are just vermin, right? They can be eradicated. That's so. I don't know if it answers your question, but yeah, that's been central. Thank you.
Thank you both so much for this wonderful conversation and rich set of reflections. Thank you.
Thank you.